My name is Francesca Scopetti, and this is Case Study 4 Presentation. Ammonia production in our bodies comes from amino acid metabolism. Amino acid metabolism comes from the process of transamination followed by the deamination and ultimately excreted through the urea cycle as our urine. The enzymes glutamate dehydrogenase, glutaminase, and glutamine synthetase all play a major role in nitrogen transport. The transamination role begins with the enzyme glutamate dehydrogenase. The role of transamination is for the byproduct ammonia to be incorporated into urea synthesis in the urea cycle. We'll see this a few times with different enzymes as ammonia being the byproduct. Glutamate dehydrogenase mechanism of action is to free ammonia and NADPH. The release of ammonia is achieved by the interaction of glutamate with NADP+, and then the formation of ketoglutarate. The generation of the keto acid frees the ammonium and NADPH. In most tissues, ATP and free ammonia begin the process of glutamine synthetase to catalyze the reaction from glutamate to glutamine. Glutamine is a primary non-toxic carrier of nitrogen, and it goes into the blood and then to the liver. Glutaminase is responsible for the deamination of glutamine. This reaction releases a free ammonium that can enter the urea cycle. Ammonia is loaded by glutamine synthetase and unloaded by glutaminase. These three enzymes work together to transport nitrogen to the liver. If we remember from previous units, the Cori cycle intertwines with the nitrogen transport system, which we'll see in this next part here. In the muscle, glutamate undergoes a transamination reaction with a pyruvate to generate alanine. Alanine can then be transferred into the bloodstream and go to the liver. Once in the liver, alanine undergoes transamination to pyruvate and is a substrate for gluconeogenesis, where glucose is produced and shuttled back to the skeletal muscle. The amino acid pool is constantly changing regardless of fasting or eating state of the human body. In fasted state, we have protein catabolism occurring, releasing amino acids going into the liver. Once in the liver, the amino acids are deaminated and the carbon skeletons are used to produce glucose. We first saw with this patient their high initial blood ammonium levels that rapidly rose, leading us to conclude that this patient has a deficiency in her ability to dispose of ammonia in her urea cycle. In this individual, laboratory results showed that the marker of orotic acid was elevated, but the level of citrulline was normal. The most likely deficient enzyme in the urea cycle would be arginase. The least likely enzyme impaired would be the carbamyl phosphate synthetase 1 or ornithine carbamyl transferase. It would be least likely that either of these enzymes would be impaired as they help produce citrulline, and citrulline levels are normal. So the deficiency would have to come from a later process, in that the end process where the enzyme arginase shuttles ornithine back into the mitochondria. I believe thymine, cytosine, and uracil would become essential with the deficiency in arginase, as elevated orotic acid is also characterized as pyrimidine synthesis deficiencies. I'm unsure about this correlation. I made it through reading the textbook, but I could not find much information on what amino acid would become essential with the deficiency of arginase. I just came to this conclusion through reading the textbook. The next case study, the best explanation for the presentation of gout in this person is due to decreased urinary excretion of the uric acid. Gout caused by uricemia targets the xanthine oxidase enzyme, which decreases uric acid production, increasing the total urine plasma in a person with not leaving the human body. This response is what we saw in the patient's lab results: a low urinary acid reading. High plasma urate reading relates to either lower excretion or higher synthesis. Uric acid is the end product in purine metabolism. The other choices are not consistent with the data, as the overproduction of purines and decreased salvages of purines would both elevate urinary uric acid. Reduced salvage of purines relates to the deficiency in the enzyme hypoxanthine guanine phosphoribosyl transferase. Synthesis of purines related to the metabolism of triglycerides, which accelerates the uric acid production. The production of uric acid is dependent on purine ingestion, which I will now discuss how the purine ingestion has affected this patient when they made the switch from diet soda to regular soda. The relationship between diet and uric acid production is not fully understood according to the study that I read. What has been discovered is that having a larger waistspan and BMI puts patients at greater risk for insulin resistant and leptin production, which both produce uric acid excretion. Both reduce uric acid excretion. It suggests that patients with hyperuricemia stay away from purine-rich foods like red meat, seafood, beer. There are other ways that will increase uric acid production that we are seeing with this patient's intake in fructose. Urate concentrations increase with sorbitol, sucrose, lactate, and methylxanthines. Once sorbitol is absorbed, it is converted into fructose in the liver and increases uric acid production. The patient switching to real sugar maximized fructose metabolism, which elevates the degradation of purine and reduces uric acid excretion, which we saw in this patient's lab panels. The role of high fructose rich food and drinks is not fully understood, but it has been seen to influence uricemia. It is important to take necessary action to reduce risk factors for hyperuricemia and gout. If left untreated, patients are at a higher risk factor to develop hypertension, diabetes, and affecting osteoarthritic joints. In this study, it stated how diet modifications tend to actually be less effective than, so it said how just modifying diet alone tends to be less effective. It's recommended that diet modifications and drug intervention is the best treatment. So the best treatment is to combine both drug therapy and diet restrictions, most importantly, given weight loss to manage hypertension. Potential impacts of only restricting red meat, seafood, beer, and other high-source purine foods does not seem to be the best treatment in the presentation of gout. When these modifications are coupled with a urate-lowering drug, that seems to have the best impact on managing chronic gout. Thank you for your time and a wonderful semester.